Neuromuscular junction. Let's start over here in this picture, and you can use a yellow highlighter to outline a motor nerve. Now, where is that motor nerve coming from? It's coming from the spinal cord, or it could even be coming off of the brain in a cranial nerve. So a spinal nerve or a cranial nerve. And what happens is that the spinal cord will have a lot of cell bodies, motor cell bodies in the ventral horn, and then those all converge together to go out one spinal nerve. And then that spinal nerve will go to a particular muscle. But within that spinal nerve then are many axons. So many axons of neurons. That. And you're going to have both directions going, both sensory and motor. We have some. So actually, let's call it. I called this a motor nerve, but actually this is a mixed nerve, isn't it? Because I'm putting sensory going that way. So a mixed nerve is going to contain sensory and motor. So sensory going from the muscle, and then motor going to it. So we'll use pink going to the muscle. So this pink line is one motor neuron. And once it gets to the muscle, it might branch, like in the rectus femoris, it might branch 1,000 times to control 1,000 different muscle cells. And then here's another neuron, and it is maybe branching 1,000 times. And you might have 800 of these neurons going to one muscle, and each of them is controlling maybe 1,000 muscle cells. So that's kind of mind-boggling, right, to think about it on that scale. But now let's go even smaller by far and look just at the neuromuscular junction, okay? So now we're going to look just at one of those potentially 1 million neuromuscular junctions that is in the rectus femoris muscle, just for an example. Okay, so here's an axon terminal. That should look pretty familiar to you this quarter. And you know that the action potential is coming down this direction. So the action potential started in the ventral horn in that motor neuron cell body, and it continued all the way to the end. And so you should also know that when that action potential reaches the axon terminal, that new channels will open and calcium will rush in. So this is the axon terminal. And let's use a blue pen for calcium. So calcium will rush in when the action potential reaches the axon terminal. And then we can use pink for the neurotransmitters inside of the synaptic vesicles. And in this case, you should even know exactly what neurotransmitter. There's only ever one neurotransmitter that's involved at a neuromuscular junction, whether that is for a skeletal muscle or for a smooth muscle or for cardiac muscle. Actually, that's not true. I misspoke there. So the skeletal muscles are only and ever going to have acetylcholine, but of course you could have norepinephrine on the heart. Okay, so acetylcholine is going to be released across the synaptic cleft. In the case of a skeletal muscle. Then let's put some receptors in purple. And then these would have to be cholinergic receptors, and specifically they're nicotinic cholinergic receptors if they are on skeletal muscles. And those neurotransmitters diffuse across the synaptic cleft. And bind to cholinergic receptors on the postsynaptic membrane. And then that causes sodium to rush in and depolarize the entire muscle cell in both directions. And the entire muscle cell will become depolarized. When it becomes depolarized, then calcium is able to rush in and cause the muscle to actually contract. So we call that excitation, contraction, coupling. Now, an important structure in understanding that is the T-Tubule. I'm going to use a yellow highlighter for that. Here and here. T-tubules are interesting invaginations into the muscle cell. What they do is allow ions such as calcium to rush into the muscle cell, even all the way down here. Let's say the receptors are way up on the top of the cell. Even the very bottom of the cell will be able to immediately contract in response to excitation. So the purpose is to allow the depolarization and contraction to occur all throughout the muscle fiber. In cardiac muscle cells, there's even connections between each of the muscle cells that you know are called intercalated discs. So between intercalated discs and T-tubules, cardiac muscle cells really beat as an entire unit. In skeletal muscle cells, they do have the T-tubules, and that allows the entire muscle cell, at least, to contract all as one unit. So let's go ahead and write this over here. Once acetylcholine binds to the receptors, the entire muscle cell depolarizes, and it happens in both directions. And T-tubules are critical for a speedy spread of depolarization to all parts of that particular muscle cell. Okay, next let's take a look at the sarcoplasmic reticulum. You hear that sarco word again, right? Sarco means flesh. But what organelle does it sound like from general biology? Sarcoplasmic reticulum. You're right. Sounds like endoplasmic reticulum, and that is what it is. It's just that because it's in a muscle cell, it gets its own special sarco name. Notice how close the sarcoplasmic reticulum is to the T-tubule. That's important. I'll tell you why. So the sarcoplasmic reticulum, or the SR, releases calcium when the muscle cell is depolarized. So because of the T-tubules, all of the muscle cell can become depolarized, and then immediately sarcoplasmic reticulum is alerted to that and releases its calcium all amongst the actin and myosin. And as you'll find out more and more, calcium is one of the essential ingredients for actin and myosin to overlap in a contraction. So that calcium will bind to a protein called troponin. And that I have, so here's in my little diagram here is actin, and then this is a troponin, actin, and then here is a myosin head, and then the thick part of its filament. Okay, so it binds to, calcium will bind to the troponin, and when it binds to the troponin, it will allow myosin to bind actin. So in other words, if it weren't for calcium, myosin could never stick to actin. And if it can't stick to actin, it's certainly not going to be able to pull on it to cause an isotonic contraction or hang on it to allow an eccentric contraction. So after calcium floods the area due to depolarization of the cell, then the myosin head can do what it's been wanting to do, which is bind to its beloved actin. And then I want to call what's happened on this page excitation, contraction, coupling. The depolarization, let's put that down here. Excitation, contraction, coupling. What this is depolarization is connected to the mechanical movement of myosin on actin. And then to wrap up this page, I would like to say what the three elements are of a neuromuscular junction. You could probably look here and see, right? You've got this, which is the axon terminal, this space, which is the synaptic cleft, and then the muscle fiber itself. Those are the three elements. So the axon terminal plus the synaptic cleft plus the muscle fiber.